Okay, we're live. So we're going to go into chapter 11. I think I've already sent you the video about the muscles. So we're going to look at the nerves that innervate the muscles. So we're going to be looking on page 382. Here you're going to have a lot of organizational thinking. How is the nervous system set up? Various groups, various components, and so forth. So got a lot of little pieces we're going to put together, and then we'll talk about maybe in the next video the action potential again. So you've been exposed to it with the muscle, but we'll get back to it again. A couple of little details that are peculiar to the nervous system. So as you look on page 382, you see the fellow there in figure 11.1, and you notice up top it on the left it says CNS, which you know is central nervous system, and then within that group you've got two structures, two gross structures, gross meaning large, not microscopic, but you got the brain and the spinal cord. And then you look over to the right and you see the purple box says PNS, and that's your peripheral nervous system. All the nerves that come out into your fingers and your arms and your scalp and your ears, your face, your lips and tongue and so forth. They're outside of the cranium. They're outside of the vertebral column. Remember that the cranium and the vertebral column protect the central nervous system. Once those nerves get out, they're no longer considered central nervous system. They actually come from there, but they're going out to the periphery. That's why they call it that. And so you have cranial nerves, as you see, cranial nerves. Well, one that you might know a good bit about is if when you go to the dentist and he goes back in your back of your mouth and he puts that needle in there, he's working with what we call a trigeminal nerve, which will innervate your lips and parts of your face and your teeth and so forth, tongue. So those are still called cranial nerves, but then they're peripheral, but they're associated with the cranial structures. And then the peripheral nerves, those spinal nerves that come off and go out to your shoulders and your arms, your feet, and so forth, your back. So you want to understand that composition. You want to understand that structure. Now, as you come down underneath that figure, 11.1, it basically breaks it down for you. So you can read about the central nervous system. And if you look in the column, if you look at the sentence just past the brain, you see it says enclosed completely by the skull. The brain is composed of nervous tissue. This remarkable organ is about 100 billion neurons. 100 billion neurons organized perfectly so that messages from your eye, messages from your ears, your taste buds and so forth, the cells in here that pick up odors and so forth, they're part of the 100 billion neurons here. And then you see, if you come to the second column, it talks about the foramen magnum. You probably know this because you studied the parts of the skeletal system. And so you got this big hole in the bottom of your head, bottom of your skull, and out comes the spinal cord, which we'll talk about a little bit later. And you know that big hole is called the foramen magnum. So magnum, some of you like Magnum PI, I guess, but whatever. Anyway, that's where the spinal cord exits the cranial cavity and then goes down through the vertebral column. Then you come to what's called the peripheral nervous system, and we've already talked about that. So again, there's just a little blurb there on cranial nerves being peripheral and spinal nerves being peripheral. Coming down into the second column, you see functional divisions. Functional, not just positional or anatomical divisions, but functional. And as you see the bold print terms in there, you come down to the second column, and you come to the second sentence in that paragraph. However, all these tasks or functions generally belong to one of three types of neurons. You see sensory, integrated, and motor. And your sensory functions pick up information from the environment, whether it's acoustic or visual or olfactory, which is a big word for smelling something. It picks up information. So some of the nerves are sensory in function. They have another term. Yes, we'll look at that. Look in the next paragraph. It says sensory input is gathered by sensory or afferent neurons. They have the afferent division, and afferent means that the message, whatever that might be, the warmth of the sun on your skin, you're aware of that, and then you start thinking, man, it's getting kind of hot out here. I need a hat or whatever. I need a hat all the time. But those neurons pick up the environmental situation, and they send the message to the central nervous system. You're made aware that you're getting hot. You know, somebody who's got leprosy, you've probably heard of that disease called leprosy. It can destroy the neurons in the cooler parts of your body, like your nose, your ears, your fingers, and so forth. And people can get burned holding something that's too hot for them, but they can't feel it because the bacterium that causes leprosy destroys sensory neurons, at least on the periphery, not necessarily in your GI tract or anything like that. Some of those bacteria can create some big problems. You might learn about that in microbiology. Anyway, you see it's sensory division or afferent division. So they bring messages to the central nervous system, and then you see the motor, and the next bold print term, or efferent, and that carries action potentials, nerve impulses, electrochemical signals. They come out of the central nervous system and go to some sort of effector. We'll mention that again. An effector is a hand, a muscle. Of course, it's made of a lot of muscles in there, but that's an effector. The muscle is going to contract, or it might be a gland, and that gland would produce a substance that is necessary for that moment. So you have sensory division, you have an efferent division. So again, as you look at the next paragraph where it says PNS sensory division, you see it says sensory information is first detected by structures of the PNS, peripheral nervous system, called sensory receptors. Now, we've already talked about that a little bit, haven't we? Like the nerves in your nose. So you know you're smelling brownies versus chili. So you have a sensory component there. You come down to the bottom, you see it says somatic sensory division. That's going to be your body, your skin, so forth. And then you see come down the next bold print term, visceral sensory division. And look at the viscera. See, that's why I don't call smooth muscle in the sense of where it's located. Viscera means it's in the organs. And look at the organs they list. Your lungs, your heart, your stomach, your intestines, your kidneys, and your urinary bladder, and so forth. So they pick up stomach aches, your bladder's full, things like that. So they would send that information just like the somatic sensory would send messages from your skin and so forth to your central nervous system. So you come over to page 383, and you look at this lady who's about to kick a soccer ball, and you can see that she picks something up visually, goes to the brain, And then a message comes down the spinal cord. Spinal nerve comes out, activates the muscles that she wants to activate in order to kick the ball to somebody else or kick it into the goal. So you've got these pathways in your body. Isn't that amazing? We start from one cell, and as we grow into trillions of cells, cells know how they're going to be neurons, and they connect with each other and make these pathways. It's like your house. You've got it wired. You flip a switch, on comes a ceiling fan, on comes a light or something like that. Just amazing how that comes to be. So we have now a mixture of a PNS, which is sensory, and then motor division. Sensory, she picked up on it, and then motor division, you look to your right of that picture, and you see there, it says it consists of motor neurons that carry out motor functions of the nervous system. So the pulses, the action impulses, action potentials move out of the central nervous system, move to the peripheral nervous system. And in these cases, if you look through this little paragraph here, you see it says motor output traveling from the brain and spinal cord through cranial and spinal nerves may be used to control the contraction of muscle or the secretion of a gland. That happens when we go home. If I go home tonight, when I go home tonight, my olfactory system is going to pick that up, and then a message is going to come down, and I'm going to start salivating because I smell what my wife is cooking, and it's preparing me to engage in the ingestion of that meal. So these glands that produce saliva to moisten things up while I'm going to be eating, those are called effectors. And my muscles, the masseter, you remember that? The masseter muscle is going to be working this jaw, and that masseter is a effector. It cause an effect. So you come down past that little paragraph where it says effectors. And you're already familiar with that because you've heard about homeostasis, which I questioned you on last time in the third test. The somatic motor division has neurons that send messages to muscles. You come down to the visceral motor division. Notice how they use that, and they're using it correctly. It's your organs inside. The visceral motor division is called the autonomic division. Now, don't call it the automatic. Autonomic means it works independently of your conscious control. Autonomy speaks of independence, and so these particular certain nerves are going to do what they need to do, and you don't have to think about it. Like producing juices in your stomach, making the glands produce more mucus so food can slide more easily through there. You don't have to think about that. All you get to do is eat the good food. I wish you probably had a little more stomach to put it in. But anyway, in that particular little paragraph there, it talks about the visceral motor division, but it's also known as the autonomic or independent nervous system. Now notice, we've already said you don't have conscious control over it, so you go back to these terms here in italics. You come down about 5 or 6 lines and you see involuntary versus voluntary. Okay? Nice diagram below. You want to know that arrangement. There's nothing hard about it, but it's going to take a little bit of time for you to get that picture in your head so you can reproduce this illustration. Say, over here we've got the peripheral somatic sensory system or whatever. This over here is the motor division, and it sends messages to muscles or whatever. Instead of receiving, it sends messages. All right, so let's leave that page. And we're on 384 now. We're at module 11.2. Notice the learning outcomes. Look at those, and as you go through the material, you'll have those somewhat in focus, in view, and that's what you want to know. How do you describe the function, the structure and the function of each component of a neuron? Well, here we're going to come over to page 385. We're going to look at this neuron on page 385. This is just a generic type of neuron, but you do want to know about the anatomy. Now, you contrasted back in chapter 10 the skeletal muscles, long muscles, multinucleated, with say a liver cell. And the liver cell was sort of cuboidal, or you could talk about a urinary tract cell, in your kidney or something like that. And they have certain components that are common to all of them, like a nucleus and so forth. But then they can be in different shapes to carry out different functions. So as you look at this neuron, see a nice pretty picture there. Before we get into that, look over on page 384. Look at the bottom of the first column and look at the second paragraph. And you see in about 1, 2, 3, 4 lines down, you see the word neuron. Now, you already knew that because we talked about it back in chapter 4, I think, when we talked about tissue. And you see it's excitable, and it's responsible for sending and receiving messages, action potentials, impulses, nerve impulses, electrochemical signals. So there's one type of cell in the nervous system, the neuron, and it says the other type, you see there, it calls them neuroglia. They pronounce it neuroglia, which is okay. So either way you want to do it, some people say neuroglia, others say neuroglia, no problem. The term there, glia, is a root word for glue. So the neuroglia tend to glue nervous tissue together. They hold it in place. That's why they name these particular cells that we'll get to in a few minutes. So you look at neurons in the second column, and you see the bold print term. They're responsible for sending and receiving action potentials. Now, look on page 385, and you want to know the parts of the neuron. So you see there is a cell body, bold print there. These are the 3 basic structures that you expect to see in a neuron, the cell body. And then you see the dendrites. Dendrites receive information. And then you look to the right and you see this structure, long, curving structure, got some purple things on it that kind of look like pigs in a blanket, don't they? And that's called the axon. So when you think about the 3 basic components, you think of the cell body. And we're going to go into a little deeper detail in just a minute. And then we're going to go, you look at dendrites. They're the receiving structures. Messages come into the cell body through those. And then you look at the axon, and the axon sends a message out to some sort of an effector, or maybe to another neuron. It doesn't always go to an effector directly, but it can hook up with another neuron, the dendrites of another neuron, and then send the message wherever it's needed. Usually you have one axon, and then you can have a number of different dendrites. So as you look at the picture, starting on the left-hand side, circle the mitochondria. A lot of activity going on in this mouth right now, right? In my respiratory system and up here. And so you use a lot of energy in those neurons. So the mitochondria must be there to produce ATP for us. Then you see, let's see if they label it, they call it Nissl bodies, ribosomes in the rough ER. Ribosomes, rough ER. What does it sound like it might be making? See how you can jump back into chapter 3? Review those chapters. Review the content of exam 1, exam 2, and now exam 3. Nucleus, got a cell there. The only cell that doesn't have a nucleus when it gets mature is the red blood cell. And it made room for hemoglobin in there. We'll learn that in the next semester. Now, as you look at the center and you see the line pointing to the nucleus, look over to the right. You see that little green structure? Figure out what that is. You know it. And you can go back and look at a regular generic kind of cell and the organelles that are in there. We're looking at organelles now. So that's a general term for that. Then you move a little bit to the right and you see axon hillock. That's where the cell kind of narrows down and becomes like a base plate for this structure called the axon. And that axon is the yellow structure that you see going away. The purple structures, you see there are a myelin sheath. We will talk about them a little bit later. Some of them don't have it, some do have it. It's got its place, right? So you see the axon, and then if you follow that long little curve, you come back and you see telodendria. And I think you probably had already been exposed to that. But notice it says axon terminals, and that would be where we had, in the case of the muscle, that would be where the myoneural junction is located, with the axon terminal, the synaptic bulb, the motor end plate, and so forth. Okay. So you see processes there on page 385 down at the bottom of the second column. They transmit electrical impulses toward the cell body. You already know that. You go over to page 386, and you see the axon in the first column, sometimes called a nerve fiber. Some of them can be very long, from your hip area down to your legs or your feet, pretty long. It carries a signal away from the body. Now, away from the cell body. So we've said that several times. Now, come on down to the third paragraph, and you see the third paragraph says the plasma membrane envelops the axon. The plasma membrane is called the axonema. Now, we started out with a cell membrane, didn't we, back in chapter 3, I think it was, with phospholipid bilayer. And then when we got to the muscles, we called it a sarcolemma. So the membrane that covers the axon is called the axolemma. At least there's some consistency there, right? When you see axo, and it's not dealing with the muscle, it's dealing with the axon, that extension of a neuron. Now, in terms of the second column where you see classification of neurons, bottom of page 386, we're not going to get into the multipolar, bipolar, pseudo, unipolar, and so forth. We're not going to do that. But what we want to notice is on page 387, we're going to look at the functional classification. And so we have the sensory, which we've already talked about that, the motor in the second column, but then you've got these interneurons. Sometimes they're called association neurons, as you see. And they're found, for the most part, in the CNS, or the central nervous system. So when we receive a stimulus, it comes in, and then these interneurons direct it to various parts of our central nervous system so that the whole body responds, in some cases, to a stimulus. Okay? It's kind of like thinking a bit. And you do have some thinking going on in there. Now, as you come to the bottom of the second column on 387, you see structural groups of neuron components. Now, in the CNS and PNS, The neuron cell bodies can be grouped, just gathered together. Notice what it says. The cell bodies of neurons are typically found within clusters, a group, okay? It's what you don't want to do now, right? With COVID-19, you want to social distance. And you see it says most of the cell bodies of neurons are in the CNS, and those clusters are called nuclei. We typically think of the nucleus as being inside the cell, nothing wrong with that, but here they choose to use that term to describe a cluster of cell bodies in the central nervous system. When you get outside, they don't call them nuclei. They call them ganglia. You have a ganglion here and here. You have probably gone to the dentist and maybe needed some work done to your teeth. And sometimes he's gone all the way back over there on one side or the other. And he has injected an anesthetic. And that anesthetic is picked up by that ganglia and the nerves that come from it. And it numbs your jaw and teeth. Makes you feel kind of slow and you're kind of talk all day. But anyway, we want you to know about nuclei. We want you to know about ganglia. Okay? You'll hear more even about ganglia when you get into microbiology. Okay, you look at quick check. What are the functions of the cell body, the dendrites and so forth? And don't worry about the multipolar, bipolar, and all that, but then the function. Functional differences, okay? Now, we got some basic knowledge about the neuron. What I want to do now is go into the neuroglia, or neuroglia, if you want to call it that. And these are cells that don't transmit impulses, but they support the neurons, which do transmit the impulses. So you see there in that first 2 or 3 sentences, it brings the word glue out in front of you. Holds the neurons together. And you can see a group of them down below. It's a real nice picture, figure 11.6. And so figure 11.6 points out 1, 2, 3, 4 types of neuroglia or neuroglia. And if you look at, there's a little bit of an explanation of what they do. They don't all do the same thing. In some respects, they protect you in a way. We'll see that in just a second. So as you look over in the second column, you see astrocytes. And of course, that makes you think of a star, doesn't it? And you can locate an astrocyte down here. Look at what they do. You're not going to get everything, but look at the first thing under astrocytes, and you see anchoring neurons and blood vessels in place. Stuff doesn't flop around in here, even in your stomach and stuff like that. In your abdominal cavity, things are anchored so they don't go the wrong way around. So it anchors neurons and blood vessels in place. Come over to 389. Come down that first column. You see it says it assists in the formation of the blood-brain barrier. That's a barrier that is intended, as we understand it, to keep microbes out. It does that a pretty good job of doing that, but it can also be a little bit of a problem when we get an antibiotic. If we do get some sort of a bacterial infection in the brain, it's hard to get a good bit of antibiotic into the brain because of the blood-brain barrier. So you see it says astrocytes facilitate the formation of a protective structure called the blood-brain barrier. It ensheathes capillaries and induces their cells to form tight junctions. Remember, and there we are again, right? Back in chapter 4, in terms of tissues, you got the desmosomes, the gap junctions, the tight junctions. You can't get away from it. That's why I tell you, read the old material. Keep it swirling around up there. Make connections, because we don't forget about it, because they're right here. Okay? So make sure you go back and review the content of exam 1 and exam 2. And you see it says these tight junctions render the capillaries virtually impermeable to proteins and polar compounds. Okay, so let's come down to repairing damaged brain tissue. Sometimes that happens. People get concussions, right? That's right. Now let's come down to oligodendrocytes. That's a mouthful, isn't it? Oligodendrocytes. Say that about 10 times. That'll wake your tongue up in the morning. And you can read about them. It says the flattened ends of some of these processes wrap around part of the axons of certain neurons. And what they wrap around is collectively called myelin, M-Y-E, myelin. It's a lipid. Got to have lipids, and we'll talk about that a little bit later. But anyway, it wraps it around some of the structures. If you would look on page 388 and look at the bottom of that illustration in the middle there, you see it says oligodendrocyte, and you see it looks like little pigs in a blanket, doesn't it? Y'all ever eat pigs in a blanket? That's what you call them, right? But good, dip them in some good sauce. But anyway, that's an oligodendrocyte. And as you look back on page 389, you see it says it wraps around, it produces concentric layers of plasma membrane that are collectively called myelin. And that wraps the axons. Now, we will talk about that function, why that is occurring. Part of it is to hold things in place, but the other part is it helps messages move more quickly. We'll get to that a little bit later. And then you come down under oligodendrocytes, you see microglia. And so, again, glia, okay? And if you look at the last 4 lines, it talks about phagocytosis. So if something gets in there, we've got some phagocytes, and they will try to eat up, whether it's a virus or a fungus. You know, you've heard of the brain-eating amoeba. Some of those cells will try to kill that amoeba if you get it. You usually pick it up through going up your nose and up way up here in the top. That's why you don't want lake water and stuff to, or even drinking water to get up your nose. Had a guy who his friend used a neti pot. You ever heard of a neti pot where he sort of flushes out the passageway up there? He didn't boil the water before he ran that water through there. So in our water, we have sometimes this amoeba that can create a problem. It doesn't hurt to go down our GI tract. It's got to come in this way, through your nose, up at the top, if it's going to create a problem. You can swallow it, doesn't bother you. You'll learn about that in micro if you take it. Okay, so we know that the microglia are there to protect you. And you come down to the ependymal cells. You remember the cilia that we talked about on some of the cells? Well, look on in here and you'll see ciliated neuroglia, second line, called ependymal cells. You and I have holes in our head. We have cavities. That's probably better. So you've got these cavities in your head, in your brain, and there's a fluid that flows through there. And it gets on the outside, and we'll talk about it a little bit later, but it's like a little cushion between your skull and your brain. Okay? So those cells with the ciliated components, these ependymal cells, they are always waving like this, and they are moving that liquid along. We'll get into that a little bit later. They call that fluid cerebrospinal fluid. Cerebro, brain, spinal, your cord up and down. Okay? So those are neuroglia that are found in the central nervous system. You look on page 389, second column, you see neuroglia in the peripheral nervous system. Got a couple of examples there. And you have a nice picture up top. You can see Schwann. I like swan, okay? It's got a Schwann. Got to practice to get that right. That's a person's name. I think it's a German. But anyway, you can see that they can surround axons, and we'll talk about them a little bit later. But you see it says larger axons of the PNS. This is by Schwann cells with a myelin sheath that has structural and functional properties nearly identical to the myelin sheath in the CNS. Different kind of cell, but it gets the same job done. And then you see little satellite cells that surround the cell bodies of neurons that are outside of the central nervous system. Okay, they appear to enclose and support, which you see it says they appear. We do not have it all down. We don't know all that goes on. We know enough to help some people sometimes, but you are going to be growing in the knowledge of this. It's one of the fun things about it that I enjoy. You learn something new, and I go, wow, how about that? But it takes a long time and a lot of visual, a lot of observation. And you go, oh, I thought it did this, but it doesn't do that. That's science. Good science is always backing up a little bit and saying, well, we saw this, but we didn't see that. So don't ever think science has got it all figured out. Don't forget to do the quick check down here. What are the functions of astrocytes, microglia, and so forth? And then the number 6, the neuroglia of the appendable cells. Okay. Let's look on page 390. We've got a few more minutes here, 390. And you see where it says, in that first column says myelin sheath. Now notice, come on down where you see the third line, composed of repeating layers of the plasma membrane. What's plasma membrane made out of? Phospholipids. Keeps coming back on you. The plasma membrane of the neuroglial cell, so it has the same substances as in any membrane. The main components, various lipids, and it gives you some examples of lipids. Oh, there's that horrible molecule cholesterol again. We don't have it. We got a problem. The trick is homeostasis. Get the right amount. See, we need it. And phospholipids, and you see there it says some unique lipids. I was kind of wondering what that what's it called? Prevagen. One of the things happening to me is I'm forgetting stuff. Oh, it's frustrating. And I see these people that have been on TV for 3 to 5 years or something like that, maybe longer than that. And it's supposed to help you remember things. I might just try that out. If I come in goofy one day, you'll find out. Don't go eat the Prevagen. But anyway, we'll see how it works out. So as you look at the picture, I want you to you see the dendrites come down the cell body, and I want you to look at the little purple things that look like the bread around a hot dog. Internode. That's called an internode, between nodes. Move to the right and you see nodes along the A. That's the little bare areas. Now, just to get you ready for thinking about this, not today, we're going to cut it off pretty quickly. I want you to think about how an electric wire, a copper wire, has insulation on it. Just think about that. This lipid that surrounds axons, it's like insulation. Okay, don't want to break that insulation in our houses. We can have a fire or something. But you see the nodes, that's the little area where it's uncovered by the lipid. And then right below the nodes of Ranvier, you see Schwann cell. that wraps around. You got a number of layers there. And so you want to know those structures. Now look at the difference here. You have below the oligodendrocytes, and you see they can do 3 or 4 cells at one time. But the Schwann cell does one section of an axon. Then another Schwann cell takes up where that one left off, leaving those little gaps. Now you think, oh no, that's not good. Maybe so in your house, but it's got its place in the physiology of the human body, and we'll get to that a little bit later. So you see the difference between the myelin sheath in the central nervous system and the myelin sheath in the peripheral nervous system. Okay? So those axons would be called myelinated. Look over here in the second column on page 390, and you come down and you eventually run across myelinated axons. And you see below there, unmyelinated. Now, this is getting you thinking about this. I told you there's going to be insulation, right? Notice it says conducts action impulses 50 to 150 times faster than an unmyelinated axon. I did a little surfing, I guess, finding out some things about this. And we can have action potentials that can travel at over 200 miles an hour. Pretty quick, isn't it? Okay, so you know about the unmyelinated axon, you know about the myelinated axon. Here's something that'll help you when you look at pictures. Look on 391. I want you to look at the second column, and you see this little paragraph. In the CNS, not CNA, that's a certified nurse assistant, right? In the CNS, you can actually see which regions of the brain and spinal cord are myelinated and which are not myelinated. How do you recognize that? In sections of both the spinal cord and the brain, okay, we're going to see darker and lighter colored tissue. Now it says the color difference reflects the distribution of myelin. The lighter colored areas are composed of myelinated axons, and they're called white matter. The other axons that are not myelinated are called gray matter. Now, some of you may have had a parent who was medical or something like that, and they might have told you, use your gray matter. And what they're saying is think. That's basically what they're saying. They're saying think. So when you see gray matter, that's where, at least up here in the skull, up in the brain rather, that's where thinking goes on. Then messages get sent from the gray matter down to some sort of effector. Okay? Well, we're going to knock it off for the day. I'll probably get back with this maybe tomorrow or something, but when we come back on the next day, page 393, we'll look at the electrophysiology of neurons. You already have a foundation in that. When you read and I've got to send you the Word document on the muscles, but if you've seen any of the videos, we talked about the resting membrane potential, the depolarization, the repolarization, the reversal of the polarity of the charge, and so forth. Well, same thing. It just happens to be in the neurons as opposed to being in the muscles that we covered back in chapter 10. Okay, I'm going to knock it off here, and I'll get with you. I'm getting your tests graded. Some of you did real well. Some of you came up quite a bit. I'm really proud of you. It really does my heart good to see you do that. I know you've put some effort into it. Don't quit. 2 more tests, and that will be, let's see, I can tell you on this video. We'll start the 4th test on the 23rd of July at 8 a.m. Okay, and you'll have through Saturday till 6 o'clock. That'll be the 25th. Okay, all right. So you guys have a nice evening, and I'll get back with you again. We'll finish this chapter. Lots of fun. Okay.